Hello, judges. I hope you are all doing well and staying safe during this COVID-19 pandemic. My name is Gavin Zhao, and I am a junior from the Academy of Medical Science Technology in the Bergen County Academies. My project aims to determine, is tau phosphorylation in Alzheimer's disease linked to oxidative stress-induced mitochondrial dysfunction? To start off, Alzheimer's disease, or AD, is one of the most common neurodegenerative diseases afflicting older people, and is mainly characterized by memory loss, cognitive impairments, and changes in personality. Nearly 1 in 10 Americans over the age of 65 are afflicted with AD, and it is the only disease within the top 10 leading causes of death in the US that cannot be prevented, cured, or slowed. Alzheimer's has multiple factors, including 2 main protein dysfunctions, amyloid beta 42 and tau. Tau proteins are usually regulated by site-specific phosphorylation, but in AD are abnormally phosphorylated and form tangles, becoming unable to maintain normal microtubule dynamics. As you can see in the image to the left, healthy brains contain stabilizing tau proteins, which results in a healthy neuron. However, when the tau proteins phosphorylate and form tangles, the microtubule begins to disintegrate, creating a diseased neuron. Next, oxidative stress is well known to cause mitochondrial damage. Additionally, aging in AD is known to transform tau proteins into phosphotau and tau tangles. However, while both healthy mitochondria and normal tau proteins are present in healthy neurons, the relationship between mitochondria and tau after experiencing oxidative stress is currently undefined. The main objective of this project can be divided into 2 parts. The first is to confirm the connection between oxidative stress and mitochondrial dysfunction. This would allow me to be certain that my model is correct and that this oxidative stress could be linked to my next step, which is to establish a link between oxidative stress-induced mitochondrial damage and tau phosphorylation. This would allow for a greater understanding of AD's pathology and give me options for further research. I hypothesized that, with increasing concentrations of H2O2, levels of phosphotau in cell circularity will increase. Levels of phosphotau increasing would indicate that oxidative stress increases tau phosphorylation. Increased circularity indicates that the cells are rounding up and coming off the plate, which means that the cells are dying and suggests mitochondrial damage. Briefly, I'd like to mention a couple assumptions that I made. First, hydrogen peroxide is assumed to induce oxidative stress. Additionally, SHSY5Y neuroblastomas, which are human nerve cancer cells, were assumed to be a good model for human neurons. Lastly, MitoTracker Red was assumed to specifically stain mitochondria. While none of these points were proven by my experimentation, I had the confidence to make these assumptions based on their product descriptions and general knowledge of what these compounds do. Here is a brief summary of the materials, equipment, and facilities involved in my project. Materials necessary for the culture of SHSY5Y cells, poly-D-lysine and glass cover slips for confocal imaging, hydrogen peroxide, or H2O2, to induce oxidative stress, MitoTracker Red CMXRos for staining of mitochondria, formaldehyde for fixing cells, and a tau phospho ELISA kit. The incubators, confocal microscope, and microplate reader from the Stem Cell Lab and Nanostructural Imaging Lab at the Bergen County Academies were used. This is a flowchart showing the process of my project. I started off by culturing SHSY5Y cells, and after plating them and treating them with H2O2, my project took 2 main experiments. The first was staining cells with MitoTracker Red, fixing them, and taking pictures with the confocal microscope for eventual measurement of cellular circularity with image J. My other experiment was to do a tau phospho ELISA using those same cells. Data analysis for both of these was performed using a one-way analysis of variance test, or an ANOVA, to determine statistically significant differences among multiple groups. I'd like to begin by showing these images that were taken in the confocal microscope. These are SHSY5Y cells that were not treated with H2O2 and stained with 500 nanomolar of MitoTracker Red for 15 minutes in a 37°C incubator. Afterwards, the cells were removed from the MitoTracker solution and fixed for 10 minutes in formaldehyde. Figures A and C are images of the cells without fluorescence. The cells are clearly flat, indicating that they are sticking to the plate and healthy. Figures B and D are images of the cells with fluorescence. As you can see in figure D, the red fluorescence is grouping up in distinctly tubular patterns. Since MitoTracker Red is known to enter mitochondrial membranes and stain them red in living cells, these tubular patterns are mitochondria. These images served as my control model and made it clear that healthy mitochondria are slightly tubular and that the dye is distinctly separated within the cells. Additionally, healthy cells are flat and stuck to the plate. Next up are pictures of SHSY5Y cells that have experienced oxidative stress. Cells were incubated with 5, 10, 20, and 50 micromolars of H2O2 for 4 hours in a 37 degrees Celsius incubator, and afterwards stained with MitoTracker Red in the same procedure as before. Figure A is a Z-stack of a cell that was treated with 10 micromolars H2O2. A Z-stack is essentially a picture of the same cell at different depths. The cell is rounder, and the MitoTracker is spread out more evenly throughout the cell. Since MitoTracker Red is supposed to be in the membranes of mitochondria, if it's evenly spread throughout the cell, then the mitochondrial membranes might have burst, indicating mitochondrial damage. Figures B to E are images of the same cell that have been treated, are groups of cells that have been treated with H2O2. As the concentration of H2O2 increases, the cells lose the edges found in figure B and end up as these circular blobs, like in figure E. As mentioned previously, circularity can suggest mitochondrial damage because the cells are likely to be experiencing cell death. If the cells aren't healthy, then their mitochondria are probably in a similar state. Because of this, I thought it would be valuable to measure the cells for cell circularity. So, I took the representative images from each well and measured circularity using image J. As you can see from the graph, circularity increased from well to well as H2O2 concentrations increased. Additionally, the ANOVA test gave a p-value of less than 0.05 for the 50 micromolar cells, which means that my results are statistically significant when compared to the control. This supports my hypothesis that with higher concentrations of H2O2, circularity increases. And because of what I explained earlier about circularity indicating unhealthy mitochondria, these results link oxidative stress with mitochondrial dysfunction. After taking the confocal images, I performed a phospho-tau ELISA to measure levels of phosphorylated tau. The SHSY5Y cells had the exact same treatment as before, with the same concentrations of H2O2. Then I graphed a standard curve using the standards from the ELISA kit, which created figure A. The standard curve is an estimation of protein concentration based on a serial dilution of a known concentration of protein. By plugging the values from the ELISA into the standard curve equation, I was given an approximate amount of tau, as shown in figure B. Because of the abnormally low tau levels, repeated ELISAs need to be done. My project so far has allowed me to do a couple things. For one thing, I established human SHSY5Y cell models with appropriate fluorescence mitochondrial markers. I also experienced ELISA procedures for the measurement of phospho-tau. Overall, my results linked increasing H2O2 concentrations with increased cell circularity, which suggests that hydrogen peroxide-induced oxidative stress leads to mitochondrial damage. Additionally, the results suggest that manipulating mitochondrial membranes, such as the mitochondrial mechanisms, such as the possible relationship between tau and mitochondrial damage, allows for further understanding and protection against Alzheimer's disease. Based on my results, there are a couple of steps that I need to take in the future. The immediate step that I am planning to take is to just repeat my experiments. I'm planning on repeating my ELISA and doing tests to adjust the cell lysing procedure. I hope to establish an ELISA method where I can induce oxidative stress and still have cells left over to run an ELISA with. For instance, I could give the cells a period of recovery time after treatment so as to obtain more cells for lysing. Next up, I plan to use my lab's electron microscope to take images of a single mitochondria after treatment and measure for quantifiable differences in shape and structure. This would allow me to be completely certain that oxidative stress and mitochondrial damage are correlated by looking at direct images of the mitochondria. After all, cell circularity only suggests mitochondrial damage and does not guarantee it. So far, my project has been focused on understanding AD's pathology, but my ultimate goal for this project is to find a way to prevent, slow, or even cure AD. Based on my results, I hope to use mitochondrial therapies to slow AD onset. The simplest way is through testing various drugs and gene therapies to see if tau phospho levels increase or decrease. My other method is localized small molecule therapy, which includes increasing cell content of mitochondria and modulating oxidative stress using antioxidants. By applying some of these therapies, I hope to protect the mitochondria from damage, thereby slowing down AD onset. This project was done in the Bergen County Academies Stem Cell Lab and Nanostructural Imaging Lab. I'd like to thank Dr. Robert Pergolesi and Dr. David Reeves for mentoring me throughout this process. Thank you, judges, for your time.